I'm Brandon.
I'm Ray, and we're chemistry graduate students in the Goldsmith lab at the University of Wisconsin-Madison. We study single molecules, and this is our lab.
In order to think about how small a single molecule is, let's talk about water. 1 drop of water contains 10 to the 21 molecules of water. If each of these molecules were blown up to the size of a basketball, they'd be large enough to create a whole new basketball planet. In other words, there are more molecules of water in a single drop than there are grains of sand on Earth. So in order for this to be true, we must realize that molecules are very tiny. So how could we possibly see them and study them? The answer is with light. So the molecule we'll be working with today is called rhodamine 6G. It's about 10 times larger than a molecule of water. But still, that's nowhere near large enough for us to be able to see and study like a microscope. So what we use is a technique called fluorescence. So this rhodamine 6G, this is a vial of our dye, and you can see that it has this orangish, pinkish color to it. And this is just what we see under regular room light. But watch what happens when we shine on it with the UV light. You can see that our rhodamine 6G emits a bright yellow color. And it's this yellow color that allows us to see and study these molecules.
Alright, so we've said we want to use light to study single molecules, but how do we do that really? Well, our lab uses a laser system and some fancy optics to get the light where we want it so we can actually use it. The optics are on this table here that I'm going to walk you through, but you'll notice it doesn't look like a normal table at your house. And when I press down on the edge of it, you hear a really loud hiss. That's because the surface of this table is actually supported on a thin cushion of nitrogen. Any small movement in this room, like, for example, construction going on downstairs, or cars or trains passing outside, has the possibility to make vibrations that could move these optics ever so slightly. We want to cushion these optics from even the slightest vibration that might change their position. The laser we're going to use for this experiment is a green laser, because as we said, our rhodamine 6G is orange to your eye, meaning it absorbs green light. You can see that our laser comes out here, and we move it through a couple of mirrors to get it into this fiber optic cable. The cable moves it around and brings our beam where we want it, and makes it look more how we want it to. We have additional control with this pinhole, which lets us cut out some of the beam if we want, or let all of it through. This next optic, this funny looking wheel, is a series of filters that lets us control how bright the beam is. You see, if I move it to a lower filter, I can make the beam a lot brighter. Or if I move it to a higher filter, I can make it dimmer. Next we have a shutter, which just lets us shut off the beam right here if we ever want to do work further down on our table without the beam being in the way. Once our laser comes out here, we bring it into a telescope. A telescope is a series of lenses, which are pieces of glass that are specially made to change the size of a beam. You'll see that as I move my card from one lens to the other, my beam expands, getting bigger. And then when it hits the second lens, it's the same size moving across the table. We call that collimating. Now that our beam's all the way over here, it's going to hit a series of mirrors. Mirrors are just like the ones you might have in your bathroom at home. And they're going to bounce our beam back and forth. Again, bringing it where we want it to go. These little knobs inside the mirrors also give us very precise control over where exactly our beam is placed. This set of mirrors here, these two, form a periscope, which is going to raise our beam from this level to this level. That's the height we need it to be to get into the back of our microscope. And this last lens here is a wide field lens, which just expands our beam really broad. This lets us fully fill up the back of our microscope and see as many molecules as possible.
So now we've gotten the laser laser into our microscope so we can look at our sample. Before we talk about this microscope though, I want to talk to you about a microscope you might be more familiar with. This microscope is called an upright microscope. It has a lens piece and then an objective which focuses light onto our sample, which is on the sample plane here. In contrast, this microscope is called an inverted microscope because the objectives, housed here, are underneath the sample plane. The reason that we do this is because we want to look at our sample in solution. So here we have a cover glass with a solution of our sample on it, and our objective pushed up against the bottom of the cover glass. This allows us to see the sample without dipping our objective into the solution. In addition, there are a number of different optics inside the microscope that allow us to focus the beam onto our sample so you can actually see the green light coming out of the objective, and other different manipulations we want to do to the light. For instance, this piece here is called dichroic. Now, dichroic is a fancy filter that blocks out specific colors of light. This allows us to look at our sample and only see the light coming from our sample instead of also seeing the green light from our laser. So, we can either look at our sample through the eyepiece, or we have a camera attached to this setup that allows us to take videos. We're going to show you one of these videos now. The video you just saw was a sample of rhodamine 6G. How would you describe it? Some people describe it as twinkling stars. Is that the same as what we see in the vial? We don't see any twinkling in the vial, just a constant light. Why might that be? Remember how many molecules were in a water droplet? In this vial, there are tons of molecules. So even though they're individually blinking, at any given point, a whole bunch of them are still giving off light. This is why we're interested in looking at a single molecule. It lets us see behaviors that you might not see in a larger sample. This sample is like having a conversation with 100 people. You're not going to be able to understand what anyone is saying, and there's going to be a lot of noise. But if you have a conversation with just one person, you can understand what they're saying. We want to have a conversation with molecules one-on-one, so we can listen to what they have to tell us. Here in the Goldsmith lab, we also study single biomolecules, like proteins, and single catalysts, which are chemicals that make reactions go faster. By looking at these chemicals, we can gather new insights for how they work. Plus, it's really cool to be able to see a single molecule. We hope you enjoyed this demonstration. Thanks for watching.